Good morning, my name is Susana Chalarca Lopera and today I'm going to talk about the impact of the junction addition molecule A on the asthma. But first we have to know what is asthma. Asthma is a chronic disease that affects children and adults. The airways that carry air to lungs become narrowed to the inflammation and compression of the muscles surrounding the fine airways. The symptoms of asthma could be coughing, wheezing, dyspnea, and chest tightening. We also have to know what is the additional molecule. Additional molecules are functional receptors that are expressed on the cell membrane and actively participate in multiple pathological phenomena. It also participates in the migration and activation of leukocytes in immunosuppression and inflammatory response. Now, the general objective. The aim of this study was to elucidate the role of gene A in a mouse model of asthma and to determine the blood levels of gene A in asthmatic patients. Now I will start with the materials and methods. The first one is Western blot. That is an immunoassay based on the antigen-antibody reaction and detects specific proteins. It would recreate the use of gel electrophoresis to separate the proteins from the sample. This technique was used to separate long proteins found in mice. The second technique is cell culture. That is a technique that allows the maintenance of cells in vitro, preserving to the maximum their psychological, biochemical, and genetic properties. With this, it was possible to maintain normal human bronchial epithelial cells in the best conditions and then expose them to dust mites. The next method is ELISA. That is an immunoassay that can be used to detect everything from antibodies, hormones, and enzymes to viruses and bacteria when an immune reaction to disease has already occurred in the patient's body. This method was used to quantify the levels of gene A in plasma samples of patients with asthma. The IHC is a method based on enzyme-linked immunosorbent reaction using antibody to detect tissue cells antigen. This method was used to stain mouse lung sections and identify the amount of gene A. Results. So, 3 seeds. We can see on the top that the lung tissues exposed with ova have an increased filtration inflammatory cells. And in the bottom, we can see that the same cells expressed with ova have an increase of global cells. The 4A, we can see that expression in low dose is significantly higher in ova than control mice. And for 6, we can see that BJNQ and BRQ have a protein level higher in ova than the control mice. Now for F, there is a significant increase of gene A in the lung tissues mice that were in contact of ova. 5A. In this, we can see the gene A proteins have increased in HDM cells compared to control after 4 hours. And 5.6, we can see that BJNQ and BRQ have increased their level proteins in HDM treated than control at 4 hours. Discussion. So, the first author, Bar S et al. Said, in these cells, gene A, CO1, occluding and clotting proteins are important indication of epithelial barrier function included in the lung. They agree with that. Kakogianos and et al. Said, TJ composition and integrity are altered in inflammatory disease and during tumor growth. They agree with that too. We saw Kipi et al. Said, The increased constitutive expression of gene A in PUX7 receptor knockout mice may have a protective effect against bleomycin-induced lung injury. That was correct. So, my conclusions. The first one is the gene M molecule could be described as an excellent biomarker of a pathology such as asthma. In tests such as Western blot and AHC that belong to molecular biology, it shows that this area allows an effective diagnosis to rule outside pathology and in this way to be able to treat effectively those who suffer from this disease. And the second one is the impact of additional molecules in different pathologies could help to find ways to change the prognosis of this disease, using the LISA technique to identify which are the antibodies that could be used to treat this pathology. Thank you so much. That's all.